Chanitra Thuvajit from Mahidol University and my co-chair Dr. Goramit Supapipat from Chulalongkorn University. Welcome back to the second session for this today morning scientific program. I'm very happy to introduce our next speaker, Professor Jacques Galipeau. He's Associate Dean for Therapeutics Development, University of Wisconsin, School of Medicine and Public Health, University of Wisconsin, Madison. And Professor Galipeau is expert in the mesenchymal stromal cell therapies and engineer synthetic cytokines for immune disorders and cancer. He is also a founding director of University of Madison Program for Advanced Cell Therapy. At this place, he has kindly let me observe his facilities for 2 times, and I am very exciting and learned a lot from his facilities last 2 years. And Professor Galipeau is also immediate past president of the International Society of Cell Therapy. So today I'm very happy, my pleasure, very exciting to meet him in Bangkok, and he will give us a topic of clinical application of mesenchymal stem cells, current trends and future directions. So everyone, please join me in welcoming Professor Galipeau.
So thank you so much for this kind introduction. I'd like to thank the organizers, in particular Professor Harinkorn, for your graciousness and wonderful hosts, and my first time in Bangkok, and it's absolutely lovely. So delight to be here. I'm Canadian by origin, so I'm as pure as Canadian snow. I have no disclosures, overlaps, or any concerns, so that's why the French name, I'm French-Canadian. So, mesenchymal stromal cells. All of you have mesodermal progenitors in your bone marrow. They're vanishingly rare. They represent maybe 1 out of 100,000 nucleated cells in your marrow, but they play a really important role biologically as niche cells that support your hematopoietic stem cells. That's their job in life. But they also have another job. They're gatekeepers of marrow myeloid cells. Did you know your bone marrow has the largest reservoir of monocyte macrophages that are really important innate effector cells involved in both resolution of inflammation and regeneration? So they're niche cells in the bone marrow, and their job in life is they support predominantly hematopoietic stem cells and lymphopoiesis because they produce morphogens. So a long time ago, in the early 70s, folks were interested in developmental biology, and it was discovered that if you took bone marrow and you just put it in a petri dish that was TC treated, that the mesodermal progenitors would attach and form colonies called CFUF, fibroblasts. And these could be differentiated down mesodermal lineages, bone, cartilage, fat. And those were used as tools to understand sort of developmental biology of bone. But what was quickly discovered is the culture-adapted mesodermal progenitors deployed a lot of their niche cell functionality. And they produced a lot of morphogens, chemokines, and cytokines that were immunomodulatory in nature. And the thing about MSCs is, you got a young graduate student in a room, you give them like 1,000 bats, and you get 30 cc's of their marrow, and you can grow their cells, a gram of cells in 2 weeks. That's 10 to the 9 cells. That's a clinical dose. But you keep on going. And you can grow from that same donor 1 kilo of cells. And these are not transformed. And so then people figured, hey, is this a solution looking for a problem to fix? They have all these functional attributes, and you can grow them under GMP circumstances. They're very easy to grow. And then the idea would be, what was discovered is because of their niche properties, they could be deployed for either regenerative medicine or immune modulation. So moving along, the first sort of big in vivo data that suggested that they could be immune modulatory were experiments done in Osiris, which was probably the first commercial entity trying to develop MSCs as a pharmaceutical based in the US. This was in the early 2000s. And there what they found is, there's a skin allograft model in non-human primates. Skin is super immunogenic. So if I take skin from you and try to give it to Natia, Natia is going to reject it within 11 days or 7 days. They did this in monkeys. And if they gave the monkeys MSCs IV, it delayed the rejection of the allogenic skin. So that was suggestive that giving MSCs as a transfusion product was immune modulatory. Move forward a number of years. And Katerina LeBlanc, who's a bone marrow transplanter, a bit like this morning's first speaker. And in Europe, you can do a lot of compassionate use one-off with a one-person IND. And she had a young boy that had an allo transplant, bone marrow from his mother. But he was dying from acute graft-versus-host disease, diarrhea, which is the white dots, and hyperbilirubinemia because graft-versus-host attacks your bowel and your liver. Super sick. Katerina grew the MSCs from the boy's mother and gave the boy a transfusion of 2 million cells per kilo of mom's MSCs. He went to remission of his GVH. There was a relapse. She gave a second infusion, and the patient went in remission of his acute graft-versus-host disease. This is a case report. Lancet never, ever publishes case reports. Why? Because this was a Lazarus effect. There was literally no precedent of severe GVH going to complete remission following any treatment. So this was the face that launched 1,000 ships. And then, of course, then companies got involved, and companies do what companies do. They're promotional. I get it. You have to be. You got to raise funds. You got to, make your shareholders happy. And they went very far. Now they were promising MSCs could cure everything. And, they didn't put hair loss in there. I wish they had, but, that's a different conversation. And their whole spiel was they could take cells from one donor and manufacture 10,000 doses from a single donor. This is not my slide, this is their slide. And off the shelf, and there you go, and you get it. So their first big use case was built upon that case report. Graft-versus-host, they figured, that's the home run, that's gonna work, So they did a clinical trial looking at they call it the remestem cell. There's a WHO way of, if it's a stem cell, anyway. So and what they did was, it was a placebo-controlled study of giving 2 million cells per kilo twice a week for 4 weeks in a row, 8 doses. If you had steroid-resistant graft-versus-host disease. Bottom line, they did not meet their primary endpoint. Giving the MSCs was no better than giving a sugar pill if you had steroid-resistant GVH. And this was published as an abstract in 2010. It took them 13 years to publish the paper in 2023. But that's because Osiris went bankrupt between then. Well, not quite. They gave up the technology. But, what they saw was, when they did subgroup analysis, children did better than adults, especially if they had liver or gut involvement, not those with skin only. So they went forward and did another clinical trial I'll come back to. So how do MSCs work? This is where I come in. So mesenchymal stromal cells, as I told you, produce a lot of morphages, but if you focus on their anti-inflammatory properties, MSCs, especially if you stimulate them with an inflammatory cytokine like interferon gamma that would be made by T cells, will upregulate endoleamine deoxygenase as well as upregulate IDO. And IDO converts tryptophan to kynurenine, and this upregulation of IDO matters. And what I'm showing here is data from our group where we looked at different normal volunteers, and what we showed was the magnitude of IDO upregulation that Natia would have versus you would have is not the same. You will all upregulate IDO, but the magnitude is epigenetically affected. So all depending upon the donor you use to make your 10,000 doses, if that donor is not a good IDO inducer, that's a bias against you. So that was one thing. The other thing, when you grow MSCs in vitro, passage 1 to passage 12, this is barcoding. You have basically a polyclonal population at the onset for the first few passages, but you get clonal impoverishment. And often, if you expand it too much, you have an oligoclonal population. Most academic studies use early passage cells within 2 weeks. Most industrial studies use stuff way out here. Big difference as well. What about freeze-thaw? So again, the bean counters, the MBAs, and the company that want to make an ROI on the commercial investment are saying, okay, you have to do allogenic, and you have to freeze it, and you ship it out like fresh frozen plasma to the hospitals. They thaw at the bedside, and you infuse everything, including the cryoprotected, the DMSO, like we do for autologous stem cell transplants. So the question we asked, is thawed cells the same as live cells? So if there's one thing I want you all to remember, stem cells, MSCs, T cells, NK cells, iPS cells, fresh is better than frozen. Who would ever offer their partner sushi they put in the freezer and the next morning thaw it out and give it to them? Okay, so any grad student that's done cell culture in this room will tell you that when they take cells in the freezer and they put in the culture, they know the cells are all messed up for a day or so afterwards. They know that. They're not going to do any experiments. They're going to wait a day or two till the cells are happy and they're growing and all is good. But what was being done in clinical trials is they thaw the cells and they infuse them no later than 4 hours after thawing. Do you think those cells are optimal? No. Oh, okay. Don't say that to Osiris, though. So we showed that post-thawing, an X and a PI, so this is live cells, there are very few dead cells. I mean, if you freeze cells correctly and you thaw them, they're alive when you thaw them. They're not dead, but they're messed up. And how are they messed up? This is the MSCs from 3 normal volunteers that we thawed and we tracked by RNA heat shock protein transcripts. They undergo a reversible heat shock response. This is a universal phenomena. Any cell, fibroblasts, HeLa cells, iPS, NK, T, name it. Thawing is an injurious effect which launches a heat shock response, and the heat shock response changes the metabolism, but it's reversible. You put them back in culture, refresh them overnight, and this heat shock response goes away. So, first generation products that were made by industry were oligoclonal, senescence, and heat shock. Other than that, they were perfectly fine. Why did the clinical trial not work? You're going to say there are hundreds, thousands of papers looking at MSCs and murine models. I know I published a lot of those papers. In 99.9% of those studies use syngeneic, fresh cultured cells. Not allogenic, not thawed. So basically they went, finally Osiris ceded their technology to Mesoblast. Mesoblast went ahead and repeated a clinical trial, but this time only in children. They excluded adults and they excluded the low-grade GVH, and they showed that there was a clinical difference. And finally, the FDA in 2024 granted the first marketing approval of an MSC product in the US, which is a big deal. Now, granted, we're in Asia, there are products approved in South Korea, in India, in Japan, and I acknowledge that, but getting an approval in the US, it was a really big deal to put wind in the cells. So, but this is, I would say it's an exhausted approval after almost a quarter century. We need to move beyond that because this does work in preclinical animal models. So what about the mechanism of action? I told you that interferon gamma upregulates IDO, it also upregulates PD-L1, but he also, and these factors enumerated here, these transcripts, COX-2, IL-6, you can read them from yourself, have all been validated as being necessary to mediate anti-inflammatory effects in recipient animals based on genetic knockouts of either the effector or the receptor on the host side. This is validated. This is not hocus pocus. And all of these act predominantly on monocytes that become polarized to an M2 functionality make IL-10. The key is MSCs don't act themselves, they act via monocytes. Is that true in vivo? So we tested that hypothesis. So our preferred go-to acute injury model is a mouse DSS colitis. DSS is soap. You put soap in the drinking water of the mice, they get toxic colitis. You take the soap away after 7 days, they recover. So we gave MSCs during the soap water phase, and you can look if there's an impact on weight loss. But what we did was we pre-treated the mice with clodronate. Liposomal clodronate is basically taken up by phagocytic cells, and it kills all the phagocytic cells, but spares all the lymphocytes. So it's a pharmacological way of depleting macrophages in a normal animal. And this will work in any vertebrate, let it be a fish or a rat. So when we gave soapy water, the mice lose weight, and then they gain weight again. If we give mice MSCs, their weight loss is less and they recover more quickly. But if we gave MSCs to mice that got pre-treated clodronate, actually their outcome was worse. This tells us MSCs work by interacting with host macrophages, and it's the host macrophages that deploy the salutary effect. So how does this work? Well, there are clues. I told you that MSC progenitors, at least those from the marrow, exist in the bone marrow. And if you give LPS to an animal, the progenitors, mesodermal progenitors, start making CCL2. These are transgenic mice where CCL2 is linked to GFP. And you know when the MSCs in the marrow upregulate CCL2, what happens? That leads to the marrow macrophages going out of the marrow space. And you have a swarm of macrophages going around, and they will accumulate in different tissues. And we found that CCL2, that indeed is the case, where they're coming from. What we found was if you give MSCs exogenously, don't forget 10 million cells per kilo, single dose, is the equivalent of your entire body content of mesodermal progenitors. But when we give those, because of what they secrete, they mobilize marrow macrophages, they go out, they go to the liver, goodly, spleen, lung, peritoneum, normal mouse. But if it's an injured mouse, the macrophages go to the sites of injury as well. So now you can understand how MSCs could be effective for any acute or chronic tissue injury syndrome, stroke, heart attack, acute kidney injury, you name it, because if you give MSCs, you mobilize macrophages whose job in life is to go to sites of injury, and they do their stuff. The MSCs don't go to the site of injury. So this slide sort of summarizes that MSCs produce morphogens, CCL2. There are others, and we've published a bunch of papers about this, that then affect CCR2 expressing macrophages as they're licensed to make IL-10, that affect T cells and B cells. And what gets released is free morphogens, but also EV bound. So this is how also MSCs can affect tissue is at a distance, because the EVs are their shuttle bus for them to go from A to B. MSC's version 2.0. So Mesoblast got its approval. I applaud Mesoblast for their perseverance, but they died past the goal line. You know, they haven't died. It's been a really difficult slog, but they're using primeval naked MSCs. Let's call them naked MSCs. You know, cells just grown in 2D and serum, the way that MSCs have been done for the past quarter century. Old school. I've told you that interferon gamma pre-licensing completely changes the cell attributes of the MSCs and replicates their endogenous physiology. So the hypothesis is, the arm waving from the companies is, well, you give the MSCs, and once they're in vivo, they sense the interferon gamma, and a fraction of them change their phenotype, and you know, is it maybe 1% or 2%? And when you give them in vivo IV, they go to the lung, they persist no more than 2 or 3 days. So we said, why depend upon a stochastic rare event in vivo, pre-license them in vitro in the Petri dish, and turn, Dr. Banner into the Hulk in the Petri dish? And that's exactly what we tested here. So what we did is we gave mice with colitis. So if you give them just a placebo, they lose weight, they gain weight. If you give them MSCs, they get better. But if you give them interferon gamma pre-treated MSCs, they do much better. So when you treat MSCs with interferon gamma, they also upregulate a whole, the interferon gamma response transcriptionally triggers hundreds of transcripts. But many of these have to do with immune world, and again, a whole bunch here. So we think that using fresh cells, and I would propose refreshed cells, is as good. You can bank them. There's nothing wrong with freezing. It's all about the thawing, and the thawing issues can be resolved by doing a culture refresh for 1 day. That fixes it all. Sleeping Beauty comes out of it looking great. Fresh and fit, pre-activation. So those are two different things that nobody else is doing. Autologous. You've heard a lot, MSCs are immune privileged. They're immune privileged. I can take Natia's MSCs and give them to everybody in the room, and it's like nothing. Untrue. MSCs express MHC class 1 like everything else. Worse. If you stimulate with interferon gamma, they upregulate MHC class 2. They can even behave as antigen-presenting cells. So the immune privilege is bunk. They, like anything else, can be allo-rejected. So autologous is a bias in your favor, especially if you want to retreat, because like platelets, platelets express MHC class 1. Patients after one transfusion can get allo-immunized. It's the same thing with MSCs. Local delivery, I think, if feasible, is advantageous. So I'm going to give you an example of xerostomia. So head and neck cancer, prevalent in Southeast Asia. You can get cured. Radiation, surgery, chemotherapy will cure you. But the radiation that you get, more than 40% of patients will get lifelong severe dry mouth because the radiation messes up your salivary glands. And the salivary glands aren't completely dead. They have dormant epithelial stem cells. It's just the microenvironment that supports them is completely messed up. And the idea we had, which we validated in the mouse model, is that if you inject MSCs in a radiated gland, can you rescue its function by reactivating the dormant epithelial stem cells to repopulate the ducts? And it does, and we've published that. And we put together a clinical trial to test that in human subjects. So what we do is patients that have had radiation therapy at least 2 years ago, they're cured of their head and neck cancer, they have severe dry mouth, we bring them in to take a bit of their marrow, and we grow their own autologous marrow MSCs. It takes 2 weeks. We interferon gamma treat them, we freeze them, and then the patient comes back as an outpatient. And an ENT surgeon, under ultrasound guidance, can inject the patient's own stem cell back into the salivary gland. We go through the submandibular gland. Did you know that you produce 1 liter of saliva a day? You just swallow it, you don't realize it. But all that's from 2 glands here. The parotids on the side here, that's what makes your mouth water when you see, coconut cake or, what is it, sweet rice? I forget the dessert I had yesterday. But anyway. But that's... The submandibular ones are the ones that constitutively make saliva, so that's why we inject those. They're a bit different. And we did a pilot study of 6 subjects, and only 6 subjects, and already we saw anecdotally patients improving with indexes of quality of life. You produce 1 liter of saliva, the young guys in the crowd, but if you have 0 saliva, even producing 5 to 10 mLs of saliva a day makes a difference. It's a bit like hemophilia. You don't need to bring factor VIII levels to 100% to get a clinical cure. You just need to bring it up to 5%. Same thing with dry mouth. And the thing that's, again, anecdotally, our first 6 patients, and this is published, so we had 3 patients that at onset, at baseline, had little to no measurable saliva production, but within 30 days had a detectable and even sustainable production 12 months, and now we have patients after 2 years after a single injection in their salivary gland, they're still making saliva. So MSC 2.0, refreshed and fit is better than freeze or burnt. Activate the cells beforehand, that's MSC 2.0. One could say that 2.1 would be combination of cytokines. One would say that 2.2 would be engineering the cells with an mRNA to overexpress another protein. The key, though, is, again, these first parameters. Autologous. Now, there's nothing wrong with allogeneic. Like, allogeneic is fine for one-and-done indications where autologous is not feasible, like acute stroke, heart attack. You don't have 2 weeks to grow cells. You have to use off the shelf, and that's fine because the mitigation of tissue loss takes place within a few days, and it takes a few weeks to develop an alloimmune response. So you have that window of time. So there's still use. Local effects can be done, and one could also think about systemic effects. I'm not going to go into that right now. So I've got my 29 seconds left here, so I'd like to acknowledge the team, colloquially called the Galipeans, and Jait and Raghavan aren't on this picture, but this is the team. I'd like to acknowledge Dr. Grace Blitzer and Sarah McCoy that are the clinical PIs on the clinical trial, as well as Andrea Pinati that has done a lot of the work also presented here. Randy Kimpel team, he's a radiation oncologist. He's the one who's bringing a lot of these patients. And of course, it takes a village from a funding perspective to get this done. I've had the great pleasure yesterday of seeing wonderful facilities are being built up here in this hospital in Thailand. You guys are doing world-class, you're setting up world-class infrastructures to be able to do the right kind of academic studies that we need to advance this space. So thanks to everybody who was such a gracious host to me here yesterday. And I'm in Wisconsin idea, for those of you who don't know that, it's all this altruistic thing that we can't live in an ivory tower coming up with these ideas and never go anywhere. We have to take ownership and make something out of it. Thank you so much for your attention.
Thank you very much, Jacques, for a very fantastic talk. I have to reconsider left my student for a vacation because they have to process cells, right? Okay, so any questions from the audiences, please?
Jacques, great talk. Your issue about viability, absolutely true. We noticed that similar issues with the T cells as well. We actually started to use platelet lysate recently and noticed that that problem sort of, to some extent, disappeared. Have you tried that on the MSCs and seen the difference as opposed to the traditional way of growing the cells?
Yeah, so we use human platelet lysate to grow our cells. And we actually tried everything in the freezing media to use HPL, FBS, HSA, autophagy inhibitors. We tried everything. The big thing here that happens also, I haven't mentioned, is mesodermal cells have a very intricate actin cytoskeleton. The process of vitrification, of freezing, depolymerizes the actin cytoskeleton. And that has a big adverse impact. That's what probably triggers the heat shock response, because it takes about 16 hours for it to reconstitute itself. And the actin cytoskeleton is really important. It's not just a skeleton. It's actually the highway by which transcriptional factors get shuttled around in the cell. And that's something that's unique to anchorage-dependent cells. I would propose probably free-floating cells, lymphoid cells probably don't depend as much on the actin cytoskeleton. But you mentioned T cells. T cells are fairly reasonably robust. B cells are very robust. NK cells are wimps. NK cells, they just go poof when you thaw them. So, but no, in our hands, the only thing that reliably allowed the cells to recover their cell attributes that are salutary was a culture refresh with an observable resolution of the heat shock response.
So the second part of the question is, what we also noticed that resting cells can be stored better than the activated cells. Does that happen with MSCs as well, that if you freshly take it from the bone marrow, without any manipulation, can you freeze them down? Do they recover better than ones compared to the ones you manipulated in vitro and try to expand that and then try to recover?
Well, the issue with using, the reason why nobody's ever used primary mesodermal progenitors from a marrow harvest is, don't forget I mentioned is, 1 out of 100,000 nucleated. So if you do a marrow and you do a ficol, 1 out of 100,000 of those nucleated cells in the ficol is an MSC progenitor. So it's not practical to try to fish those out unless you start doing single cell sorting. That's not going to work. So it's not practical. That's why putting them in culture, they attach, they very vigorously start forming CFUFs and they replicate promptly. That's why the field has used that as opposed to, which is a good idea, but it's just not practical. They're just not numerous enough.
Thanks. Great talk. You're a very engaging speaker. I was wondering, you talked about the loss of polyclonality during the passage process. Has there been many efforts to try and change the culture environment with cytokines or other immunomodulatory things to try and preserve that polyclonality?
Yeah, so the clonal impoverishment is only seen when you really push to the extreme, passage 12. Again, and that really has only affected the industrial developers that wish to develop an allogeneic platform. If you're going to be using autologous cells, we never go beyond passage 2, which is 2 weeks, where it's a non-issue. I mean, the loss of polyclonality, because you remember those 2 arrows?
Yes.
Where it was 2 weeks, and that was fine. It's really for a long-term expansion for allogeneic. Has anybody tried to find a remedy for that? Because nobody knows what's the stochastic mechanism by which that occurs. I think the remedy for that is something that's being developed currently in Australia, which is to use iPS-derived MSCs. A company called Synata is doing that. But there it's a completely different platform now. It's iPS cells, so there it's, by necessity, it's a clone. But they keep their spots long term, and they can expand them luxuriously. So that's sort of kind of the remedy right now that's being looked at to generate an alternative for expanded at scale allogeneic MSCs.
Thanks very much.
Thank you very much. So that's
Thank
all
you.
the time we have for the question. Thank you, Professor Gillespie, for the comprehensive talk on the MSC myth.